Vanakkam. Welcome to this course on classics in neuroscience. We were looking at neurological foundations that led to the development of neuroimaging or brain imaging. In the last video, we looked at the seminal contributions of Wilder Penfield to the field of neurology and neurosurgery. In this video, we will look at cerebral circulation and the scientific studies and the historical background that led to the development of research in the field of cerebral circulation and how one could move from cerebral circulation to brain metabolism. These two are interrelated, but it was not obvious. Now it is obvious, but at that time in the 1940s and 1950s it was obvious. There were some studies earlier, but somehow those early studies were not given the due importance and the credence that they probably deserved. So these studies and that field lagged behind by about half a century. But in the 1950s or from the 1950s onwards, this field picked up pace and we now have this huge field of functional neuroimaging. That is today. But where were we in the 1940s and 1950s, and how the work in the 1950s was critical in the development of neuroimaging as we now know it. That will be the focus of this video's content. Significant advancements in the field of brain circulation and metabolism within the brain happened in the 1950s, and that essentially led to the foundation of neuroimaging or brain imaging as we now know it. But the original earliest foundation to this was laid by the Italian scientist and neurologist Mosso. This work is reported by William James in his book Principles of Psychology, famous textbook at that time, 1890, where he said that Mosso observed that during mental activity in patients who have had head trauma, during mental activity there was increased pulsation, there was increased blood flow. Whenever the subject was spoken to, whenever the subject began to think actively or was solving a mental arithmetic problem, essentially blood flow increased, increasing the intracranial blood pressure. So the intracranial blood pressure rose almost immediately. So mental activation, thinking, doing mental arithmetic, listening or doing some mental activity essentially increases the blood flow to the brain. Today that seems obvious, but it was not obvious in the 1880s and 1890s. So how much blood would flow? That depends on the demands of the mental activity. That is, cerebral circulation follows the needs of the cerebral activity. More and more activation means more and more blood flows. Of course, you would not flood the brain with blood that is not needed, right? Just like the vasomotor mechanism that is present in other parts everywhere throughout the body, the brain also needs blood, and the amount of blood that is received by the brain depends on the amount of blood that it needs or the demand that is there. So more activation means, or more activity that is being performed by the brain means more blood flows to the brain. So through this reflex vasomotor mechanism in response to neuronal activity, brain cell activity, this observation is nearly 140 or 150 years ago. And in 1904, Mosso was one of the special invited guests together with Ramon y Cajal to inaugurate Clark University. This was also studied by the famous British neurophysiologist Charles Sherrington with a student who studied the cerebral cortex in monkeys, and they found that when there was electrical stimulation of cerebral vessels of cortical areas, there was changes in the local variations of blood supply or vascular supply. So, there are some intrinsic mechanisms. This suggests that there were some chemical products of metabolism that influence the activation of the blood vessels, essentially suggesting that the composition of blood and the amount of blood that is used are critically dependent on what is happening in the brain. So, there is a chemical composition, but also the quantity, both are dependent on the activation level in the brain. So, that means if I can somehow trace or if I can somehow figure out specific chemicals that are responsible for specific activity, and if I could somehow attach a label or a tracer to that chemical, then voila, I have essentially found a way to identify activation levels depending on the activity, right. So, that is now it seems obvious, because this idea is more than 100 years old. I have also read this in textbooks. Now it is obvious, at that time it was not obvious. These studies and early observations by Mosso and Sherrington practically were left in relative obscurity until it was much later recognized that someone should have followed this up. Of course, in the 1920s, late 1920s, John Fulton, who was a student of so John Fulton's observation was that there was an increase in the blood flow to the occipital lobe when there was visual stimulation during brain surgery. So, that means that occipital lobe is somehow responsible for the visual sensation or visual perception. Obvious today, but was not obvious at that time. So, there was an increase in blood flow to the region that was responsible to handle visual stimuli whenever there was visual stimuli presented to the subject. I think this experiment was in cats. All these early studies, Mosso's in 1880 to 1890 and in the 1890 study of Sherrington and Roy and John Fulton's study in the 1920s, 1928, were in relative obscurity until they were revived back by other scientists. Now it is recognized that these studies were milestones, were early seminal studies that led to the development of the field of neuroimaging as we now know it. If only people had pursued this earlier in the 1900s and in the 1910s and 1920s, we would have had development of neuroimaging earlier, but some things had to wait for their time. This brings us to our introduction to Seymour Ketty and his contributions to the field of cerebral blood flow. He introduced the method of using radioactive tracers to study cerebral blood flow. So, Carl Schmidt at the University of Pennsylvania was studying this, and he was joined by Seymour Ketty, who was a young instructor at that point, and together they started developing methods to study and measure the arteriovenous differences in nitrous oxide. So, you supply this gas to the patient through the mouth and nose, and you study changes in the chemicals within the blood. So, this is the study, and they used the same method to study the amount of oxygen, the amount of carbon dioxide, the amount of glucose and the amount of lactate. This is critical because if more oxygen goes inside and more carbon dioxide comes outside from a given organ, that means metabolism is happening in that organ. Of course, this is not going to help you identify regions of the brain. At this time, we are at that stage where how much oxygen is being consumed by the brain is being studied. Okay. So, more oxygen is entering and less oxygen is coming out, less carbon dioxide is entering and more carbon dioxide is coming out from the brain. What this means is that the brain is utilizing the oxygen that did not come out. It has converted that oxygen to carbon dioxide, common sense, right? That is what must have happened. So, products of metabolism is this carbon dioxide that is naturally going to be exhaled. So, this, although at that time did not help identify or localize regions of the brain, later methods in 1950s to introduce different tracers for different chemicals helped to distinguish between regional blood flow, the amount of glucose that is taken and oxygen consumption. They found out the activation levels or the activity that is happening in a given brain region and oxygen consumption and that blood flow to that region. All these are closely connected. That is, more activation means more oxygen consumption and more blood flows there. So, these are all closely connected, and this makes sense. Just like muscle activity, the more and more you use muscles, more blood is going to flow carrying oxygen, and that is going to be utilized for performing the muscle's task, which will then result as a product of this metabolism is going to result in the oxygen getting converted to carbon dioxide. Of course, glucose is being used, except the difference being that muscle activity can also happen anaerobically. It does not always need oxygen. It can also happen with oxygen, but it can also happen in the absence of oxygen. Fundamentally different approaches. This is a huge topic in itself. I will refer you to my videos in the other course, Neuroscience of Human Movement, in which I discuss muscle activation with oxygen and without oxygen. So, now fundamentally different topic, an entirely different topic, muscle physiology, which we are not interested in at this point in time. The point is that muscles can function even without oxygen, that is, can function anaerobically, but brain cannot function without oxygen. Fundamental difference. The brain consumes more oxygen than any other part of the body that is having the same weight and the same volume, for example. That is, when you compare the amount of oxygen consumed per unit volume or per unit weight by any region of the body, it will be found that the brain consumes a huge amount of oxygen when compared with the other organs in the body. Something to keep in mind. So, there is a great dependence of oxygen by the brain. So, brain appears to rely heavily to oxygen supply. This idea later led to the development of the 2-deoxyglucose activity mapping method that is used to map patterns of brain activity. First, it was developed in animals because of the traces that were used. It was first applicable or allowed to be used only in animals. Later, a different method called positron emission tomography was developed to study this in humans. And Sokolov, one of Seymour Cate's colleagues, emphasizes how Seymour Cate's low-key approach towards these studies led to the development of this field. Of course, Seymour Cate also studied neurochemistry, contributed a lot to neurochemistry, because he collaborated with several biochemists in Europe and in the United States, such as Heinrich Welch, Paul Mendel, Derek Richter, and Henry McIlvaine, and essentially co-initiated neurochemical symposia and found the International Society for Neurochemistry and the Journal of Neurochemistry, and was also instrumental in the formation of the International Brain Research Organization. Yes. So, but still there was this need to develop local cerebral blood flow measurements. So, I need to know which area of the brain is receiving more blood flow, because to me that means there is more activation happening in that region of the brain, or more neurons are active in that region of the brain. Of course, there are obvious limitations of the nitrous oxide method, because it only measures the average blood flow and the metabolic waste for the whole brain and cannot help to localize brain activation levels within regions of the brain. So, then a method to measure local CBF or cerebral blood flow was developed. This used a chemically inert tracer that could diffuse freely across the blood-brain barrier, and it used I labeled trifluoroiodomethane as a tracer because of its unique properties. This was then developed as a quantitative autoradiographic technique that could help us with localization, or it essentially achieved localization within the brain, but of course its use was limited to animals. So, this was first reported in 1955, measurement of cerebral blood flow in individual brain units of conscious and anesthetized cats. This method was used to examine or investigate the effect of visual stimulation. It was found that there was greater cerebral blood flow in the visual pathway, for example, the lateral geniculate nucleus to the occipital lobe, for example, and this was perhaps the first published demonstration of functional brain imaging. Today, there are many functional brain imaging techniques available, well developed techniques. For example, functional magnetic resonance imaging, then functional near infrared spectroscopy. There are several such advanced techniques available to study brain activation levels. Of course, these two are not exactly comparable, because the depth to which this can work varies between the techniques. You can read upon these techniques yourself, but there are advanced techniques such as the functional magnetic resonance imaging that is now very popular to study activation levels of the brain during specific activities. With this, we come to the end of this video. In this video, we looked at the historical background of cerebral circulation and how cerebral circulation and brain metabolism, and the study of cerebral circulation and brain metabolism led to the development of neuroimaging as we now know it. Thank you for your attention.